All right, good afternoon, everybody. It's like that time. So hopefully everybody saw the exam grades are posted. If you come by to review your exam before the second exam, you'll get 2 additional bonus points that'll go towards the next exam. All right, so stop by, review those. You can come by during regular office hours, or if you want to meet a time outside office hours, just let me know your availability and we'll work out a time to do that, okay? And students are allowed to come by anytime during the semester to do any exam or any quiz. I also all the bonus quizzes. If you want to see a breakdown, I can make a photocopy. I keep the originals since they're grouped, but I can make photocopies and give them to everybody in the group if they want. So you're welcome to stop by, review anything anytime. But if you want the 2 points, it has to be done before the next exam. Okay. So before we continue on with blood, we're gonna have our next quiz on Wednesday. So Wednesday we'll have a quiz. It's going to cover blood, what we've gone through last week as well as today. And we're also going to start doing some anatomy with blood vessels. So I think we'll probably wait until the next quiz with that. But as we're continuing quizzing on blood, we're going to start adding blood vessel anatomy. So there's a chapter on blood vessels. That's chapter 19. And so that's with the, there's a couple pictures right here. So let me just show you briefly what to start studying, because we're going to start getting into this as well as the cardiac anatomy on our quizzes, right? So this figure right here from chapter 19 that shows us important sites of palpation. That's important clinically, knowing where you can get a pulse. So those are important arteries to know. In terms of the detailed anatomy with the arteries and the veins, you need to know both, right? So in this book, arteries are generally colored red to indicate they're carrying oxygenated blood. Of course, that's only true for the systemic circuit, right? And that's what this is showing us, the systemic circuit circulation. So red and for systemic is going to be arteries, and then the veins in blue. So these figures here that are full body pictures for the heads and veins, that's going to be the major blood vessels to focus on, right? And so you're going to see blood vessel anatomy on the second lecture exam with the major arteries and veins. And so if it's on these 2 full body pictures or the site with this one where for the arteries that can be palpated, those are going to be fair game for the quiz and for the exam, all right? And in lecture, you're going to dissect many of these, not all of these, but many of these, both arteries and veins in the fetal pigs. And we have special fetal pigs that are latex injected to where the arterial system has red latex, the venous system has blue. So with the dissections in lab will kind of complement, but if you start studying ahead of time first with the pictures in the book of the humans, I think that will make the anatomy with the fetal pigs later on easier, okay? All right, so start studying those, and then also in the cardiac lab, where we do anatomy with the heart models, all those structures that you've been looking at in lab, we're going to have that on lecture quizzes too. So pretty much that's the definitive list of the heart anatomy for the lecture as well as the lab. So you're going to see that on both the midterm and final lab exam, and you'll see it on the next lecture exam, and then some of it will be on the final lecture exam as well. So that's really 4 exams where you're going to see all of this material again. So compared with the bones and the muscle from A&P1, in my opinion, it's not nearly as overwhelming as that, right? Just in terms of the probably sheer volume of information. So if you've mastered that pretty well from A&P1, you ought to be able to get through the cardiac anatomy and the anatomy of blood vessels without too much problem. But as always, aside from the time in class and lab, I'm always happy to go over stuff if students have questions. We can pull out models, things like that as needed. So start studying the cardiac anatomy and the blood vessel anatomy. We won't quiz on that Wednesday, but the next quiz after that, which will probably be the following Wednesday, we'll start having that on there. Okay, so it'll just give you a little bit of a heads up. All right. Any questions on any of that? Okay. All right, so back to blood. So last time we were talking about hemoglobin, and so as you may remember, hemoglobin has multiple states, right? Oxyhemoglobin is a state in which it is bound to oxygen. It's more bright red, right? But it also can be deoxygenated. So this is a reversible reaction, right? Formation of oxy or deoxyhemoglobin is reversible. That's what the arrow's pointing in both directions means. It can go either way. And that's extremely important. Not all chemical reactions are reversible, but this one is. And why is it important that oxygen reversibly binds to hemoglobin, not permanently bind? Why is it important that it's a reversible binding? Who can tell me? So where do you pick oxygen up in the body? What organ? In the lungs, right? Mainly, but the oxygen has to be, when it's delivered to metabolically active tissue, the oxygen has to be released to those tissues, right? Which is, so that at that point, the oxygen has to actually dissociate from the hemoglobin so that it can be in its free molecular form to diffuse into the metabolically active tissues, all right? So it has to be able to bind tight enough so the hemoglobin can hold on to the oxygen in the blood, but not too tight. It can't let go of the oxygen where it's needed, right? So binding is reversible. And actually how tightly it binds to oxygen depends on how much oxygen is already bound to other subunits. Remember, hemoglobin has 4 subunits. So the state of other subunits affects the ability of the remaining subunits to bind oxygen or not, right? And other gases can affect that as well. And so let's break that down a little bit. So we talked about oxy and deoxyhemoglobin. Those are 2 forms that you need to know, and you need to know those terms. This is a figure from the book chapter here, and there's a lot going on. As you can see here, this is showing the inside of a red blood cell, for example. So this is inside of a blood vessel. Here's inside the red blood cell, and so the oxygen is binding to the hemoglobin, but not only oxygen, what else binds to hemoglobin, as shown in this figure?
CO2.
CO2 is another gas that also binds to hemoglobin. So hemoglobin is used not only to transport oxygen, but it's also used to transport carbon dioxide. And as you remember, carbon dioxide is a waste product that's produced by basic cellular respiration, right? So as we consume oxygen, we're producing carbon dioxide as a wasteful byproduct, and it needs to be taken out of the metabolically active tissues. And where do we get rid of carbon dioxide? Couple ways we can eliminate. What's one way we eliminate carbon dioxide in our body?
Excellent.
Breathe it out and ventilate it off, right? So how do we get rid of this load of toxic substance to get out of our blood? Well, we have to get out of our blood. So there's a couple ways to do that. Some of it we breathe out, right, as a gas. But some of it we excrete through urine, right? So our kidneys and our lungs mainly are going to be the way that we can process these loads, right? So as the blood is flowing to our kidneys and lungs, that's how we get off of it. But how does the CO2 get there? Well, not all of the CO2 is bound to hemoglobin. Some of it's dissolved in the blood plasma directly, right? But most of it is actually found in the red blood cells where it binds to the hemoglobin. That's some of it, right? But not all of the carbon dioxide is bound to the hemoglobin. There's another way it is stored inside cells too. It becomes locked up inside another molecule through a chemical reaction. When carbon dioxide reacts with water, right, it forms this, which we call carbonic acid, right? That's why carbonated beverages like sodas are always a little bit acidic, because the carbon dioxide, when it dissolves in the water, it makes it acidic, right? So carbonic acid is a weak acid, so it's going to dissociate into free protons and this molecule. So this molecule is the other major molecule where the carbon dioxide is actually stored, locked up. Anybody know the name of that important molecule? It's called bicarbonate, right? Bicarbonate. So some of the CO2 is bound to hemoglobin. And hemoglobin is found in red blood cells, but some of it is stored up in bicarbonate. Now, bicarbonate can accumulate, this reaction can occur both in red blood cells and in the plasma outside of red blood cells, but you have more of it in the red blood cells. And what's the reason for that? Why does it happen much faster? Bicarbonate is produced much faster in red blood cells than outside of red blood cells. What is it that red blood cells have that you don't have in the plasma, for example, that facilitates production? What do you call something that increases the rate of a chemical reaction? A catalyst. So what do you call a biological catalyst? An enzyme, right? So there is an enzyme that is located inside of red blood cells. And what do we say? Enzymes typically end in which suffix? ACE. So what's the name of the enzyme that speeds up the rate of bicarbonate production in red blood cells?
Anhydrase.
Yeah, carbonic anhydrase. So because red blood cells have this enzyme that's not in the plasma, you get more of the bicarbonate production, right? And again, this is reversible. It's important that carbon dioxide reversibly bind to hemoglobin, so it has to go backwards. Where is hemoglobin going to bind to the carbon dioxide? Which organ? Or where? Right, pretty much all throughout circulation. Anywhere you have metabolically active tissues, that CO2 is going to be generated, right? So all throughout circulation. Where do we get rid of that? Again, it's going to be in our kidneys and in our lungs. Okay, so when we're transporting carbon dioxide, we don't want it in free form, right? Because it's toxic. So it's either bound with hemoglobin or locked up in bicarbonate. And these reactions can actually go in reverse, of course. Okay, so at our lungs and kidneys, that's where we get rid of the carbon dioxide, and that's where you're going to see things go in reverse. Okay, so going back to the concept of different chemical forms. Oh, I'm sorry, you had a question. Yes.
Do you say enzyme speeds up?
The enzyme that speeds it up is called carbonic anhydrase. Okay?
What does it speed up?
Oh, what's it speed up? It speeds up this rate of this reaction. So when carbon dioxide reacts with water, it forms carbonic acid. So it speeds that up. But when carbonic acid forms, it is automatically going to disassociate to some degree to bicarbonate, right? Now these can go back and forth because this is a conjugate acid base. I don't want to get into all that because that gets into more basic chemistry, but does that make sense? That's what it's speeding up. It speeds up formation of carbonic acid, which then is going to lead to bicarbonate production. Yes, sir.
Well, what does bicarbonate?
What does bicarbonate do? Yeah, so what bicarbonate is important. One, it's a buffer. It functions as a buffer. It helps resist change in pH, right? So pH is a measure of the concentration of protons. And so it's because it can go backwards. If pH drops, bicarbonate, right, acts like alkaline. It helps buffer that, right? So it helps. One important thing it does in our body is help buffer against changes in pH. So not only is it, so partly it's storing carbon dioxide, that's partly what it does, but it also is a buffer. And it's an important way that we regulate blood pH, right? And when we regulate pH in our kidneys and urinary function, that gets very important. Some cell types can excrete protons. So if you're too acidic, then you want to pee out protons, and those cells will do that. But on the other hand, other cells can release bicarbonate to have the other effect, right? So different cells can regulate protons or bicarbonate in our kidneys to regulate the pH of our urine. So the pH of your urine is not a narrow window. It can vary quite a bit, but what you need to maintain is the blood. Your blood has to have a very, very narrow range of pH. So bicarbonate is important to all of that stuff, yeah. And there's other body systems we'll talk about where it gets important, but mainly with that. Right, does that make sense? Okay. Any other questions about that? Okay, so going back to chemical forms of hemoglobin, we talked about oxyhemoglobin. That's when hemoglobin binds to oxygen. When it's not bound to hemoglobin, it's deoxyhemoglobin. But what do you call hemoglobin bound with carbon dioxide? There's another name for that. It's called carbaminohemoglobin. And that's designated by this. HB stands for hemoglobin. CO2 is carbaminohemoglobin, okay? So make sure you know the difference between oxy, deoxy, and carbaminohemoglobin. hemoglobin. So if you take just the part of that figure out, right, and focus on that reaction right there, the rate, this is carbaminohemoglobin, right? Okay. And again, that needs to be reversible. So carbaminohemoglobin and oxy and deoxyhemoglobin are all normal forms of hemoglobin. But there are some abnormal forms as well. So let's talk about some abnormal forms. One abnormal form is carboxyhemoglobin. Do not confuse carboxyhemoglobin with carbaminohemoglobin. Students commonly do that on exams. That's a mistake. Carbaminohemoglobin is normal, right? Carboxyhemoglobin is abnormal. What's the difference? Who can tell me? So when carbon dioxide binds, we call it carbaminohemoglobin. What do we call carboxyhemoglobin? It's not carbon dioxide, but a molecule that is very similar, but just a little bit different. Not carbon dioxide, but what? What is this? Carbon bound to a single oxygen, we call carbon monoxide. Carbon monoxide is very toxic, right? Why is carbon monoxide toxic? What happens if you're exposed to too much carbon monoxide? What's it going to do to you?
Suffocate.
It suffocates you, right? It leads to hypoxia. Why? Because it prevents your hemoglobin from carrying oxygen, right? Okay, so how does that work and why does it do that? Well, it turns out that carbon monoxide binds to the same place that oxygen does. So they compete for the same binding site. But unfortunately, carbon monoxide has a greater affinity, significantly so than oxygen. That means carbon monoxide tends to outcompete oxygen binding. And if oxygen can't bind to the hemoglobin, then we can't transport it in our blood to tissues, right? So that's how it can cause hypoxia. All right, so carbon monoxide competes for the same binding sites in the hemoglobin as does molecular oxygen. And what do we call those binding sites? Where in the hemoglobin does oxygen bind? Which groups? Anybody remember? What are these little red discs indicating? That has the iron in the middle of it. What do we call those structures? Hemes, right? So hemoglobin, right? You have the globin chains. It's the polypeptide, the 4 subunits, and each of those has a heme group. Hemoglobin is named for both the heme that binds the oxygen and the globin chains, which are kind of the scaffold for the heme, right? Okay, does that make sense? So that's why carbon monoxide is very toxic. And of course, where do we get carbon monoxide exposure? What's the most common source of carbon monoxide?
Smoke.
Yeah, it's a product of combustion. So cigarette smoke or combustion engine. And so that's why you don't want to be in a garage, right? With the windows open and garage door closed, right? That can be very toxic very quickly. Yes, sir.
So it binds to the same as oxygen as the other molecule for difference.
If it binds the same as oxygen, what's that?
Like, you know how we read the oxygen on the field thermometer? Did it tell the difference or does it read?
Yeah, so, right, it will affect, yeah, you call that pulse oximetry that measures oxygen saturation. And that's right, if it binds, it's going to decrease oxygen saturation. So you could measure that with a pulse oximeter. That's right, yeah. So, because the pulse oximeter is just looking at how different wavelengths of light are absorbed, which is affected by whether it's bound to oxygen or carbon monoxide or whatever, right? So absolutely, that's true, right? So let's talk about what about carbon dioxide? Why does carbon dioxide not do that, right? Carbon dioxide does not compete for binding with oxygen, right? So how is that different? So where is carbon dioxide binding? It's because it doesn't bind to the heme, it binds to the globin chains, right? And that's why carbon dioxide is not as toxic. Of course, at certain concentrations, it is, right? You can euthanize animals with high levels of CO2, and sometimes that's used as a method, right, in animal research, for example. Okay, so CO2 bound to hemoglobin is another normal chemical form of hemoglobin. And what form do we call that? Carbaminohemoglobin. Carbon monoxide bound to hemoglobin is toxic. And what do we call that? Yeah, carboxyhemoglobin, right? Now, it's also important to mention that, as with anything in toxicology, dose makes the poison, right? And so carbon monoxide, it turns out it's not always bad. It depends on the concentration. In fact, our nervous system produces small amounts of carbon monoxide, and it can be used as a diffusible neurotransmitter signaling factor. Okay, so at low levels, it does have a role in the body in cellular signaling, but we're talking about high levels exposure, which you would usually get from being exposed to combustion products, right? So, even if you don't have lethal dose from, revving your engine in a garage, even lower levels found in cigarette smoke and things like that can cause chronic hypoxia, right? So I wanted to share a couple other abnormal forms that weren't really detailed in the book. So in addition to carboxyhemoglobin, there's also methemoglobin. This one tends to be rare, but there are some rare genetic disorders where individuals have an abnormal form of the hemoglobin. In this case, they have the oxidized form of iron, right? And the oxidized form of iron won't bind to the oxygen. So in that particular case, they're going to be hypoxic because of the abnormal hemoglobin. Another example is sulfohemoglobin. So what causes these? Well, generally, so it can be a genetic disorder, or maybe you have a certain enzyme and metabolism that's not functioning correctly, and it's leading to oxidation or sulfation of hemoglobin. Some rare drug chemical exposures also can do that as well. Okay, so in addition to carboxyhemoglobin, methemoglobin, sulfohemoglobin, those are a couple of abnormal variants. And then we'll talk about variants of the alpha beta chains, such as in some blood diseases like sickle cell disease. There are also variants of that. All right, so any questions on any of that? All right, so let's talk about red blood cell formation. There is a word used to describe formation of red blood cells, and that is going to happen in which tissue, right? Where red blood cells, along with any blood cells, form. All blood cells are formed in which tissue?
Bone marrow?
Bone marrow, exactly. What type of tissue? What type of marrow specifically? Red marrow. What's the other type of marrow we have? Yellow marrow, right? So most of our marrow starts off red. Why is it red? Because it has lots of blood cells, including stem cells that make new blood. So where all blood cells come from? Wherever you have red bone marrow. Now, most of our marrow is red when we're born, but as we become an adult, most of it turns into yellow marrow. And yellow marrow doesn't contain blood stem cells. It contains mainly what? Fat, right? That's why a lot of people like to eat to suck the marrow of a bone. It's very tasty because it has all the fat in there, right? But yellow marrow doesn't contain stem cells. So if somebody has a blood disease, maybe they need a marrow transplant because you have a leukemia patient, for example, that needs the new marrow because their marrow is defective. You have to pull the marrow from red marrow. And red marrow has a more limited distribution in adults as it turns mostly into yellow marrow. So if you need to take tissue for a marrow transplant, where are some locations where marrow can be taken from? Right, hip, right. Hip is one common location. Epiphyses of the humerus, for example, the sternum. Your hip and sternum are what type of bones? So these are the types of bones that mostly have most of the red marrow. What type? They're flat, right? They're flat, right? Your hip, you could say, is actually irregular. It's kind of flat. So we talk about flat bones. We have that diploe in the middle, that kind of sandwich of spongy bone in between plates of thick, compact bone. And that's where most of the red marrow is going to be. So those are good places to harvest. But there are a few places if you need to do that. Okay, so let's talk about red blood cell development and different stages of that. So this is from figure 17.5 in your book, and this is showing the process of erythropoiesis. Okay, so the name of the process by which red blood cells develop and mature is called erythropoiesis. Right? Erythro means pertaining to red blood cells. Erythro means red, right? poiesis is formation, right? So with erythropoiesis, this formation of red blood cells, what would be formation of white blood cells? What do you think? What would you call it? What would be a logical name to call it? Yeah, leukopoiesis, exactly. Leuko means white, erythro is red, and poiesis is formation. Okay, so well, there's another table showing leukopoiesis, or development of white blood cells, which is more complex because, as you've seen from lab, there's not just one type of white blood cell like there are red blood cells, right? And really, there's actually a couple of types of red blood cells in circulation. We'll get to that just in a second. Okay, so this is showing how red blood cells develop, right? And so starting with the stem cell, the basic stem cell, what do we call those? We call them, well, they can go by a couple names. One name is hemopoietic stem cell, right? Or sometimes they're called hemocytoblasts. So hemopoietic stem cell or hemocytoblasts, right? Again, remember in ANP1, there was a similar topic where you would have to learn different stages of cells for different tissue types. Like for bone cells, you have osteoprogenitor cells, those form osteoblasts, osteoblasts form osteocytes, and so forth. So similar kind of concept here. The stem cell that you find in the marrow are the hemocytoblasts or hematopoietic stem cells. All right, so inside the marrow, several other stages happen until eventually you get mature red blood cells that then leave the marrow and enter blood circulation. So what other stages can you find in between in the marrow? So these are stem cells that could become other cells. They could become, at this point, white blood cells. They're not committed yet to become red blood cells. So these also take a different pathway to form white blood cells. But once they go into the pro-erythroblast stage, they are now committed to form red blood cells because it has erythroblast in the name. And again, just like with chondroblast or osteoblast, that term tells you it's an immature cell, right? So just like chondroblasts make cartilage, chondrocytes maintain it, same kind of thing here. The blast tells you it's immature. But in this case, it's not it is committed to be a red blood cell. Here, it still could be a white blood cell. What determines what fate it goes down? Well, there are signaling factors and hormones that interact here to determine the fate. Okay. All right. Once it becomes a pro-erythroblast, it's committed to become a red blood cell and is now going to undergo several other phases of development. The next stage, we call it a basophilic erythroblast. So it's no longer a pro-erythroblast, which is almost like a pro being an earlier stage. This is a later stage erythroblast. There's three types of later stage erythroblasts that are defined by color changes, right? The first stage is basophilic, largely, just like it was when it was a stem cell or pro-erythroblast, meaning that it's noted that it is very acidic, right? And therefore it has an affinity for the basic stains, such as an H&E, okay? So what happens is that as it continues developing, the cytoplasm becomes less and less acidic. And as it becomes less and less acidic or more basic, it starts to change color, right? Because like H&E, whether you see pink or purple, that's based on whether the structure is acidic or basic. So the next stage, it becomes less basic, more acidic. So now you have a mix of color, not blue, but now you're mixing it with red. And so now at that stage, you call polychromatic. Polychromatic means literally what? Poly means what? Many, in this case, more than one. And chromatic refers to what? Color, right? So it's multiple colors because there's red and blue, or you could say both eosinophilic and basophilic here. And then eventually it loses the basophilic characteristic and becomes mainly acidophilic. And that way, there we call it orthochromatic. Color chromatic still means color. Ortho means what? You go to an orthodontist to do what to your teeth? Straighten them. Ortho means straight or singular, right? So kind of all aligned. So in this case, it means there's only one color, okay? So those are the three stages. And why does the cell go from more basic to more acidic as it starts to develop? Anybody have an idea?
To maintain the pH level.
Well, then, no, not really, because the pH in cellular compartments is not regulated the same way it is like in blood, right? It does affect the pH, and that's what we can see, but it's not to maintain it, right? So the reason it's acidic initially because it has a nucleus, and what does a nucleus have inside of it? Well, nucleic acid, among other stuff, DNA, RNA, which is acidic, right? And not only that, you have DNA and RNA in here. RNA enters the cytoplasm, right? So RNA is acidic, and RNA is used to make proteins. So when the cell is cranking out proteins, it's more acidic because of all the RNA, right, in the cytoplasm. But what happens if we look at mature red blood cells? They don't have a nucleus at all, at least not in humans. In some animals, they do. It depends on the species, right? So, but that nucleus, even when they have it in species that have it, it's not producing all the acidic molecules like RNA, right? Because it's not producing new proteins. Because if it was producing new proteins, that requires a lot of energy and oxygen, and the cells would end up using up the oxygen they're transporting to make new proteins, right? That's why the nucleus becomes non-functional. In humans, it gets ejected out of the cell just before they leave the bone marrow, okay? So before that happens, the RNA production reduces, and that makes it less acidic, okay? That's where we can see the color change. All right, so by the time the cell leaves the bone marrow and enters the blood, it lacks a nucleus, and we say they're anucleate. They have that special shape that maximizes surface area and makes it able to store oxygen. But another stage, the reticulocyte stage is a stage right before that. Why does it get that name? Well, if you see, it looks like it kind of has little, almost like veiny structures there. That is a leftover endoplasmic reticulum, right? Rough endoplasmic reticulum. And rough endoplasmic reticulum is associated with ribosomes. So it's part of that protein synthesizing machinery that it generates, right? So red blood cells in blood mostly are erythrocytes. You can see some reticulocytes, and then eventually they mature inside the blood into fully developed erythrocytes. Both of those stages are lacking nuclei, right? So you're only going to see nuclei in the earlier stage cells in the bone marrow, okay? At least in humans, that's true. All right, so any questions about that? And so you might see on the next quiz or on the exam, need to know the different stages, okay? But break down the terms in terms of color, basophilic, straight, basophilic or blue, polychromatic, red and blue, mix, orthochromatic, straight red, right? Basically. Okay. Does that make sense? All right. Well, so what actually helps regulate erythropoiesis? There's a number of factors. I mean, there's genetic, but also hormonal, right? And mainly, there is one hormone. It's produced by the kidneys. It's not one we spend a lot of time talking about, but it's important for regulating blood cell development. So who can tell me which hormone produced in the kidneys regulates the rate of red cell production in the marrow? It's a hormone called EPO. EPO stands for, anybody know? Erythropoietin. So it's a hormone named because it regulates the process of erythropoiesis, right? It's produced in your kidneys in response to hypoxia, okay? So your kidneys actually have chemoreceptors that can detect levels of oxygenation. So if you go, up to New Mexico or Colorado, the mountains at higher elevation, you're going to have lower oxygen. That's going to stimulate your kidneys. Certain cell types can start producing EPO. Okay, so EPO is produced by your kidneys and is important for red blood cell development. So if you have something like anemia, it could be a cause of kidney disease, right? Could be a cause of your anemia, or it could be a problem inside the bone marrow, or it could be a problem elsewhere, because as you can see, many of these body systems are connected, right? But kidney disease, because of that reason, when you have kidney damage, it often causes problems like anemia and abnormal blood growth, right? And where is EPO having an effect? Right here. EPO essentially increases the rate of this first step. where it forms a committed pro-erythroblast. So if you increase the rate of this step, you increase the rate of production of all red blood cells. And so if you go up to a higher elevation, the first couple of days you might have mountain sickness as you're trying to acclimatize, right, to the new oxygen levels. But after a couple of days, your hematocrit will end up going up because your kidneys are going to start producing EPO, and that's going to increase red blood cell, right? So why is it that some athletes like to train at high elevations? It's very common, right? Although in sports medicine, I think it's a bit of a controversial topic, exactly how effective it is. Won't get into that, but what's the principle? Why would you go train at higher elevations if you're prepared?
So that you can breathe better.
Yeah, it increases your cardiovascular endurance, your aerobic endurance specifically, right? Because if you train in the mountains at a higher elevation, your kidneys are going to be producing more EPO. Your hematocrit goes up. It will increase your red blood cell count. So when you compete at a lower elevation, you have ability to carry more oxygen, right? And some athletes, of course, go farther and they do doping and stuff like that. So you've heard about doping scandals before, right? With events, and that's a problem in a lot of different sports, of course. And what's the reason for why you don't want to inject EPO to do this synthetically?
And
Right.
it like combust, like, can it expand too much and it?
So what happens, so as you increase red blood cell, you increase your hematocrit. What's that going to do to your blood? It's going to affect the viscosity. Is it going to increase or decrease the viscosity? It increases it. Blood's thicker than water, right? But if it's too thick, what happens? It clots, right? And you get obstructions. So it's risky doing that, obviously. That's why in normal sports, we're not doing this. But through training at high elevations, you can naturally take advantage of it to some extent. Now, whether it's beneficial or not, you have to ask sports scientists about that, right? So anyway. All right, so red blood cell production, the rate of production is very important. Your kidneys play an important role in that, as well as your bone marrow. So marrow or kidney disease can affect that, right? Other diseases that cause malnourishment or malabsorption of certain key nutrients can also affect that, right? So some gut diseases can also lead to anemia. What's the reason? Because in order to produce red blood cells to go through that pathway, there are a couple of key nutrients that are needed on top of everything else, right? And namely, those are going to be B9 and B12. Those are examples of water-soluble vitamins, right? So vitamin B is part of the water-soluble vitamin group. And B9, another name for B9 would be what? Folate or folic acid. Lots of it in leafy greens, right? If you're deficient for folate or folic acid, you increase the risk for neural birth defects, but you also, if you're pregnant, but even without pregnancy, it can put you at risk for anemia, for example, if you're deficient in it, because you have to have high levels of it. Also B12, cobalamin, right? Also needed. So if you are deficient in B12, you might need to take B12 injections, for example, in order to boost that. All right, and that's because they're needed at key steps for DNA replication. And for cells, when they are being created rapidly, always at a constant rate, they're going to be very reliant on anything that affects the rate of cell division, right? And why do we have to constantly produce new red blood cells? So not all cells do this. We don't do this with neurons, right? Your neurons are not constantly replicating in your brain. They're post-mitotic, right? But these cells are not post-mitotic, and they continue to replicate at high rates. We're always having to make new red blood cells throughout our whole life. If anything ever inhibits that ability to make new red blood cells, it can lead to anemia, right? But why do we need to make so many new red blood cells? What's the reason? Well, they don't live that long, right? Because they don't have a nucleus or proteins, the cell can't be kept alive indefinitely in circulation, which means once red blood cells leave bone marrow, they're only going to circulate for maybe up to 3 months. So they can do maybe 90 to 120 days, 3 or 4 months before they die. And that's because they can't make new proteins, right? And so you have to keep making them all the time in order to replace them. Okay, so you have to, because of the high rates of division, you have to have the nutrients in order to sustain that. Another important dietary factor, aside from B vitamins to maintain DNA synthesis and cell division, you also need iron, right? Of course, you have to have enough iron in your diet, or you can have iron deficiency anemia. That's a type of anemia. Why? Because iron is what actually binds the oxygen. It's part of the heme group in the hemoglobin, right? And so iron, of course, we have it in a number of different tissues, not only in our red blood cells. But when red blood cells are broken down, it liberates that iron, and something has to be done with that iron to reduce its toxicity, because free iron in any tissues is always toxic. So iron, in order to not be toxic, it has to be complex with some kind of protein, right? So we talked about carrier proteins, globulins, and plasma, for example. That's one example. Is there iron circulating in your blood outside of red blood cells? Sure, but it's complex with a globulin protein in plasma called transferrin, all right? Otherwise, it's toxic. In tissues, you have it, particularly, anybody know which one organ that's very rich in iron? Your liver, for example. Your liver very rich in iron, right? So your liver and other tissues that are high in levels of iron, they have a couple of other proteins like ferritin or hemosiderin that actually binds to the iron. Okay, so that's another problem. All the iron in these red blood cells, they have short lives, and when they die, that iron is released, and it's toxic if it's not bound to other proteins. So that's what some of those other plasma and tissue proteins are doing, is helping to bind and neutralize the toxicity of free iron. All right, so we talked about red blood cells. They live generally, for normal individuals, between about 98 to 120 days in humans. So that's about 3 to 4 months, right? And then they die and they have to be replaced. So there is always some rate of destruction of red blood cells, some rate of red blood cell production. And in order to maintain a constant hematocrit, or level of circulating red blood cells, you have to balance the rate of production and destruction. Okay, so production is erythropoiesis. It's under control of hormones and other molecular signaling in the marrow. Okay, what about control of red blood cell destruction? Right. So where does that occur? Mainly in one organ, which is in the spleen. But there's another organ that does it too. And if you have your spleen removed, this other organ pretty much takes up over all the rest of the function of the spleen for this. So your spleen, and does anybody know what other organ is red blood cells removed? Your liver, right? So if your spleen is removed, your liver can take over the function of the spleen, at least that aspect of it, okay? So we're not gonna get all the minutia here of how that does that. We will take a look at spleen later in the chapter when we get into lymphoid organs. We'll talk about spleen a little bit and look at it, right? So how is it, but does anybody know how is it that the spleen is in liver, but mainly the spleen is good at getting rid of old red blood cells? So it turns out that old red blood cells, as they age, they become more rigid. They lose that flexibility. And later on, as we'll talk about, the spleen is the only organ in the body where you have an open circulatory system. When I say an open circulatory system, who can tell me what I mean by it? Here's my alarm. I'm going to go ahead and pass out the sign-in sheet. We'll keep it going. There you go. So who can tell me what I mean by an open circulatory system? Ours is mostly closed. Some insects and things, no. Right? So ours is closed, meaning what? Where is our blood enclosed in? In our circulatory system, blood vessels, right? So closed means the blood is within blood vessels. But there in the spleen, there are actually open-ended blood vessels where the blood gets released into the general tissue of the spleen, the stroma of the spleen. Okay, so that's the only place in the body where blood will actually leave the closed circulatory system and enter the tissue spaces. Now, those blood cells have to get back into the circulatory system. And to do that, they get into these they squeeze through little crevices and leaky capillaries, a specific type of capillary called sinusoidal capillaries, right? So to get back into circulation in the spleen, they have to fit through the little crevices in the walls. And to do that, they have to be flexible. If they become rigid, they get stuck. And when they get stuck, there are a bunch of macrophages hanging out. Anybody remember what do macrophages do? Remember from AMP1? Right, phagocytosis. So when they get stuck, macrophages go and they eat the cells, right? And when they eat them, they have to break down the hemoglobin, and it gets broken down into the hemes as well as the iron. The iron and hemes have to be metabolized. The iron is toxic if it's also not complex, right? So these cells are always broken down, and there are macrophages in the liver that can do a similar thing. All right. So when the blood cells are broken down, the hemoglobin is also broken down and metabolized, right? So the globin part, those are polypeptides, are made of amino acids. Those amino acids, when they're broken down, they're going to circulate in the blood until other cells take them up to make new proteins. So the globin part gets recycled in that way. What about the heme part, right? So the heme part is also going to get recycled, the iron to some extent, right? So the heme is going to contain the iron. When it's broken down, you have the iron that then has to get complexed with a protein like ferritin, hemosiderin, or transferrin. Otherwise, it's toxic in the tissue, right? The other part of the heme, after the iron is removed, it's still a pigment, but it's a pigment that now shifts to a slightly different color of red, and we call it bilirubin. Okay, bilirubin. Rubin means ruby color, and it's red, but it's actually a little different color, red. And bilirubin is actually more what color? It accumulates in your blood, it turns your blood and eventually your skin what color?
Yellow.
Yellow, right. And so what do we call that condition? If a patient has yellow skin coloration, what do we call that? Jaundice, and it indicates what type of disease or not functioning. Not kidneys. Liver, right. Because where is bilirubin eliminated from the body? By the liver. How is it eliminated? What does the liver produce that bilirubin mixes in with to get it out of your body? This is the other name of bilirubin. So the rubin part tells you it's a yellow-red pigment. The bili tells you it gets secreted into what? What does your liver produce that is secreted in your gut? It helps us emulsify fats, but it's also an important route of elimination of things from our body. Bile. Okay, so bilirubin is a name for a bile pigment. The bili refers to its release of bile. Your liver, as we'll talk about, makes bile, and bile gets released into your gut. So anything there is going to get defecated out, right? Okay, so we eliminate iron through our feces because the bilirubin gets processed by the liver. So any liver damage can cause jaundice, right? And that's only normal when is jaundice actually normal? Yeah, infancy, right? What's the reason for that? If a newborn baby is often jaundiced and doctors don't get alarmed, why is that more normal?
Because isn't that like livers, it doesn't.
Yeah, when they're born, their liver isn't
Yeah.
kicking into full gear, right? Their organs are starting to really function because the maternal placenta has done a lot of the work, right? So it's not fully functioning, but once it functions, it starts to clear out the bilirubin, right? So that's the only place it's considered normal. But in an adult, it's always considered pathological to some degree. Okay, all right. Any questions about any of that? Okay, so bilirubin is a yellow pigment. When it accumulates, it tells you your liver's not functioning properly. You can't clear it from your blood. It causes yellowing of your skin, jaundice, right? So, but when it's functioning normally, the bilirubin, it gets metabolized. There are some other molecules here. It goes into other pigments like biliverdin. But eventually, in the gut, it turns into another pigment called stercobilin, right? And stercobilin is which color? Which color is stercobilin? Brown, right? So the reason why feces, stools, tends to have a brown color, it's that it's again that the iron, right? It contains iron in it. But iron can change color depending on which metabolic form of it, right? Or the heme, I should say, excuse me, the heme of the iron can change color based on exactly the chemical form of it. So in the gut, bacteria and other cells convert the pigment into a final pigment called stercobilin, and that's what makes your feces brown, right? We also eliminate it through our urine, right? So urochrome, for example, is a pigment that gives our urine kind of a yellow coloration, and same kind of thing, right? So the coloration of our excretions is actually due to different colors of this pigment as it's being eliminated. All right, any questions about metabolism of hemoglobin, bilirubin, any of that stuff, liver function? Okay, all right, so let's talk a little bit about disorders, blood disorders. So again, in the endocrine chapter, a big part of that is most of the emphasis is, what's the normal function, but it's also always important to talk about when things don't function correctly, that leads to disease. So when we talk about blood diseases, there's a number of different types of blood diseases. For example, leukocyte disorders involve white blood cells. Erythrocyte disorders involve red blood cells. So we'll talk about some of each and some other types of blood diseases as well. So let's talk about erythrocyte disorders. And so one example of an erythrocyte disorder is anemia. But anemia is a very general term. It's a blanket term. If you say somebody's anemic, you don't really know the cause of the problem. It could be a number of different causes, right? Until you get a differential diagnosis, for example. So anemia, in its most general sense, if somebody has anemia, all you know is what? What does that tell you?
Right.
Not necessarily low iron, because some types of anemia, you can have normal levels of iron. So if you have iron deficiency anemia, yes, that's the cause. But what if you have a different type of anemia? It may not be tied to that. So no, it's more general than iron. That can be one type of anemia, but more general than that.
Red blood cell.
Even more general than that, because there are some types of red blood cells that have normal counts. If you might be low in iron, but have the right number of cells. They just, each cell is deficient in iron. So that's not true either. Although again, there are some types. So all anemia tells you is that there's a deficiency to transport oxygen in the blood, right? Involving your blood cells, right? That's gonna lead to hypoxia. So it's some sort of blood disorder that is going to lead to hypoxia and inability to carry oxygen to cells. And there's a number of different causes. That's why it's not really a disease itself. It's more of a sign of disease. So when we look at major causes, we can think of really three categories, right? So one cause would be blood loss, right? We call that hemorrhaging, right? And so it can be quick, it can be over a slow period of time. There are different examples, but if you lose blood, you're losing not only the blood plasma, but you're losing the red blood cells, and that's what it carries the oxygen. So blood loss will cause that. Low red blood cells production can have it, right? So that's one cause, and that could be caused to exposure of environmental toxins, radiation, chemotherapy, things like that. It could be missing dietary factors, right? And then a third major cause is that you're producing the red blood cells at the right rate, but something else is destroying them. They may, when they're born, they may be too fragile, for example, and therefore they're destroyed at a high rate. And as we said, if the rate of red blood cell production and destruction is not balanced, right, it can lead to under or overproduction. Okay, all right, so let's look at blood loss and specific types. We call anemia due to blood loss hemorrhagic anemia. Hemorrhagic means you're hemorrhaging, right, losing blood, right? And that's divided generally into two categories, acute versus chronic. So who can tell me what would be a difference or maybe an example, real world example of acute versus chronic hemorrhagic anemia? In general, forget the anemia part, just acute versus chronic. What's the difference? Who can tell me?
It's like, it's not as fast.
Which one?
Blood loss?
Which one's not as fast?
Acute. No, acute is fast and chronic is slow.
Right, so acute is generally gonna be faster, chronic's generally gonna be slower. Acute means on a short term, it's about time span, and chronic is gonna be a longer term, okay? So let's look at some examples. There can be a number of examples, and we'll talk about all of them. If it's acute, it's gonna be rapid blood loss. So gunshot wounds, stab wounds, things like that, right? Or examples of acute hemorrhagic anemia. What about chronic hemorrhagic anemia? Well, there are a number of causes. Hemorrhoids, for most people, they are kind of a minor to major annoyance. They can cause pain, but also they can cause enough blood loss, they can actually cause anemia if they're not treated, right? Bleed ulcers, gastric ulcers, stomach ulcers, for example, can be a cause of that, right? Some people may have in old age, that's pretty common. You might die in your sleep, right? Because you're slowly bleeding over many days, months. And so chronic hemorrhagic anemia might be harder to detect, right? You're not necessarily going to see a lot of visible blood in your stool, but it can still, if it happens for a long period of time, it can still be enough to make you anemic over that period of time. Okay, so there's also, when we talk about low red blood cell production, that the iron deficiency anemia kind of fits in with that, although it's not necessarily the number of cells per se in that case. So an example of iron deficiency anemia, for example, the cells, it's not that there's fewer of them necessarily, but they're abnormal, right? For example, they might appear hypochromic. Hypochromic means what? What do you think that would mean?
Low.
Low color, right? Chromic's color, because it's the iron in the hemoglobin that's the pigment, right? So they might look more pale if they're deficient in iron, and sometimes they're smaller. So we can, if they're abnormally small, we can call that microcytic anemia. Microcytosis is abnormally small. Okay, so that's another example. But there are other types of anemia. In pernicious anemia, you might get macrocytosis rather than microcytosis. And that's where the red blood cells are abnormally large, maybe as large or larger than a small lymphocyte, for example, right? So let's talk about pernicious anemia. Anybody know what is pernicious anemia? What's the cause? So it turns out that and does anybody know, well, if you don't know the cause, how do you treat it? How do you treat pernicious anemia? The treatment for pernicious anemia is B12 injections, right? So because it comes back to deficiency in vitamin B12, right? So the gut is not able to absorb vitamin B12. Does anybody know why we can't absorb vitamin B12, or where does most of the B12 we absorb? What part of our gut is that going to be? Anybody know?
Small intestine.
The small intestine, especially the more distal part of the small intestine, like the ileum or distal jejunum, we'll talk about later, right? So you absorb it through your small intestine, but in order to absorb it there's another protein we have to make that's made elsewhere, and that protein is made in our stomach, right? And that type of protein, it's a glycoprotein to be specific. It's produced in our stomach, and it binds B12, and if it's not complex with B12, when it gets to the end of the gut, you don't absorb it very efficiently, right? So pernicious anemia is caused by a deficiency in this glycoprotein. It's thought to be an autoimmune disorder, similar to many endocrine diseases, where your immune system attacks these cells in your stomach, and they don't produce enough of this, so you don't absorb B12 later on. Does anybody know the name of the protein or glycoprotein? It's called intrinsic factor, okay? So pernicious anemia is an autoimmune disorder, right? Kind of like with type 1 diabetes, your immune system attacks, in that case, attacks those beta pancreatic islet cells. In this case, it's attacking cells in your stomach. Later on, when we talk about digestive system, we'll talk about different cells and their different activities, right? So when you're deficient in intrinsic factor, you can't absorb B12, and that can also cause anemia. All right, so generally the treatment is B12 injections, right? You can't take it orally. If you take B12 supplements orally and you can't absorb across your gut in the first place, that's not going to help you, right? So usually you have to inject it, right? B12 is water soluble, so you have to take it orally as a pill or inject or something like that. So you have to actually inject it every day in your muscle, right? In order to get the effect, right? Correct amount of red blood cells. All right, a couple other types of anemia, again, being a general term. Renal anemia means it's a disease associated with what organs? Renal anemia means disease with what organ?
Kidney.
Your kidneys, right? So kidney disease. Why? Because your kidneys produce EPO, and EPO regulates the rate of erythropoiesis, right? So kidney damage, we call that renal anemia, right? There's also a type of anemia called aplastic anemia. Aplastic in this context means they're not being produced correctly, and that's because something is destroying or damaging bone marrow, right? And oftentimes it is exposures, right? There may be some genetic causes, but oftentimes aplastic anemia is linked to environmental exposures, right? So things like certain drugs or chemicals like chemotherapy, right? If you know somebody that's undergone chemotherapy, they almost always will have anemia, right? To some degree. That's because chemotherapy targets cells that divide really quick, including not only cancer cells, but normal cells as collateral damage like red blood cells, right? That always have to be replaced. So chemotherapies often damage the stem cells and marrow and can cause anemia in that way, right? If you had radiation treatment for cancer, same thing. So cancer treatments being very harsh can damage the marrow. But there can be other causes as well. Some other environmental toxins and things like that also can affect it. All right. So any questions on those types of anemias? So of course, there's other categories too. So let's talk about hemolytic anemias. So hemolytic anemias are where you have high rates of destruction, right? What is hemolytic? Hemo means tells you pertaining to the blood, right? You got to think hemoglobin in the blood, and lysis. So lysis means what? Like hydrolysis. We talked about that early in AAP 1. What does hydrolysis do? If something's lysed, what happens to it? It splits. Hydrolysis breaks macromolecules with water, right? Splits the molecules. So, and when you're talking about hemolysis, you're talking about red blood cells that are fragile. And because they're fragile, they're likely to break apart or lyse, right? Some viruses lyse cells also when they regenerate to destroy cells. Okay, so if your blood cells are made in, normally, and you have normal numbers of them, but they're fragile, then you can still have a deficiency there. Okay, so this isn't going to be a problem with the bone marrow necessarily, but it's some other problem. And we won't talk necessarily about every type of hemolytic anemia, but there are two important classes that are very important for medicine because they're pretty highly prevalent. So one of those groups of diseases are called thalassemias, right? So my father always told me I was at risk and she had screamed because, I'm half Greek. And so this is Mediterranean. So this type of disease tends to be associated with people with Mediterranean ancestry, right? Another group of diseases is sickle cell anemia. So those are both. And so thalassemias are associated with Mediterranean, Greece, Italy, areas like that. What about sickle cell disease? What ancestry is most associated with it? African, right. So what is it that the Mediterranean region and African regions have in common? Why we tend to see these gene variants overrepresented in these populations. So actually having these, if you're in the United States, having these variants may not be useful. But if you're in Africa or the Mediterranean, it can actually protect you against something, because what blood disease are these regions plagued with? Malaria, right? Malaria has killed more people than just about anything, right? Bullets, right? So malaria is a huge scourge, has been for a long time. One of the most common causes of death that we know of, right? That's not natural. So well, I guess it's natural in a certain way, but so sickle cell anemia and thalassemias can be protective if you live in malaria infested regions, right? What's the reason for that? And that's why those genes tend to stay in a population. And if you live in that part of the world, it's not bad to have a copy, right? Actually, if you're talking about sickle cell anemia, the best case scenario is to have, if you're in one of those regions, have possibly one mutated copy and one normal copy, right? Because you're somewhat protected, but don't suffer all the bad effects. Why is it that this protects you from malaria? What types of cells does malaria infect to complete its life cycle? Right, red blood cells. So it's a protozoan, a type of parasite. They infect red blood cells, and so the red blood cell for them is an incubator. So if it's very fragile and breaks, then they can't complete their larval development, right? So that's why if you live in those regions, having one copy of this gene that makes your red blood cells a bit more fragile can actually be advantageous because it protects you from malaria. So we've seen that these copies of these forms of genes, there is a lot of selective pressure for them to evolve in those areas, right? So sickle cell anemia and thalassemias produce fragile cells, and they can protect you from blood parasites. But otherwise, they're a liability because they also make the cells more fragile. They can cause problems, right? And so with thalassemias and sickle cell disease, there's actually different types. It depends on where the mutation occurs. It can affect, if it's thalassemias, they're usually grouped into alpha and beta, depending on whether the mutation's in the alpha or beta chains of the hemoglobin. If it's sickle cell disease, it's generally going to be a mutation in the beta chains, but there can be different types. And generally, it's only a single mutation, a single amino acid substitution. So for example, in one type of sickle cell anemia, on the gene that encodes the beta chain of hemoglobin, there's a substitution from a glutamic acid to a valine. One amino acid changes the structure of the beta chain and the whole hemoglobin enough that it acts abnormally and causes other problems, right? So then what happens? Specifically in sickle cell anemia, when you have a sickle cell crisis, then the red cells start sickling, right? So they change from their normal shape that we discussed to looking kind of elongated and sickle shape. Think of like the sickle, on the farming implement. Okay, so what causes the cells to sickle up and under what conditions? Generally, under hypoxic conditions is mainly when this happens. That's why a lot of times people that have a sickle cell trait, which is what you call if you have one copy of the gene, or it's anemia if you have both, they often don't know it until they're doing an athletic event or something, and where they're exerting themselves and their oxygen drops enough to cause the cells to sickle up. So it's usually in the venous system where oxygen levels are lower than in the arterial system. And what happens is you get more deoxyhemoglobin, and because its shape's a little bit different, it causes it to clump up abnormally. And when it clumps up, it fills up the cells and it causes them to sickle up. And those sickled cells are more fragile, they break apart. They also tend to clump, they're not flexible, so they cause clotting and obstructions, and it can be an extremely excruciatingly painful condition because you get a lot of pain due to hypoxia in the tissues and things like that, right? So anyway, that is sickle cell anemia, right? Any questions on that? Okay, 5 minutes left. So let's talk about another disorder, polycythemia. Polycythemia is something different. That is where you have overproduction of red blood cells. So you're not, you don't have a deficiency in erythropoiesis. You're not producing fragile cells. In this case, it's really the opposite. You're producing too many red blood cells, right? Well, there are a couple different types of polycythemia. One type is called polycythemia vera, which is true polycythemia vera. If you're being truthful, right? You're speaking with veracity. That's where that word comes from. That's true polycythemia. True polycythemia is when you have a problem with the marrow itself. Generally, that tells you have bone marrow cancer, okay? Because some of those stem cells are cancerous, so they're dividing uncontrollably, and that's a primary cause. But if you have polycythemia, it doesn't always mean you have blood bone marrow cancer. It could be secondary to something else. So secondary polycythemia is where you have disease in some other area, and your body is producing more red blood cells to compensate, right? So there can be different examples, and this can be normal if you go train at high elevation. That's a normal polycythemia. Your kidneys are going to start producing more EPO to increase your hematocrit, and that's not disease. That is adapting to a higher elevation, right? But disease can cause it too. If you have pulmonary problems, you might have COPD or something like that, where you're suffering from chronic hypoxia. Okay, and so your kidneys are going to react the same way as if you go to higher elevation. They detect the drop in oxygen, and so they start cranking out more red blood cells. You can compensate for that and carry more oxygen, right? Aside from lung disease, cardiovascular disease. If you have circulation problems, that also can lead to that, right? So polycythemia, it may be cancer in the bone marrow, or it could be your kidneys compensating for a problem in some other area. All right, so our next topic is going to be leukocytes, and I think it's about a good place to stop before doing the next part. So we'll start with your next topic.